Should not take much time. Now, once meanwhile the file is downloading, I will request you all to download a tool called FastQ Dump. So once you have copy pasted the link using wget in your terminal, or once you have started downloading via the browser, you can always go. Yes, the GEO ID is GSE 205178. So, meanwhile, your data is getting downloaded. I would request you to please download SRA Toolkit, install it in your Linux system, or you can also search for install FastQ dump in Linux or Ubuntu, whatever, in Linux command line, in Ubuntu command line, whatever is easy for you. Search. It will give you a GitHub page, which will it provides all the files. If you have a Windows system, if you have Ubuntu installed, so accordingly you can download it. Just follow the instructions given here. The same installation usually would not take much time. I'll provide the link here. You can download Ubuntu version, it should work, or you can also download Windows version. Please try using Ubuntu version. But if you're working in Windows OS, I'll recommend you to download the Windows version. Or else, if you want a raw data, FastQ raw data, instead of, in case you have trouble with downloading these datas, I'll share a drive link once again. Or you can check your WhatsApp where I have shared the drive link. You can just a second. I'll uh, I'll give you a drive link where you can directly download FastQ files. In case you are facing trouble with installation of tools. so that we can continue forward with our. Please use this link and you can download. There are three FastQ files which I was able to upload in the drive. You can download them and you can proceed forward. We can proceed forward with our analysis. And meanwhile, the original SRA dump or FastQ dump toolkit analysis, this you can always make a note of it and you can always practice this later on as well. I would request you to please make a note of these commands. One second. Once you are done with downloading the FastQ files from the drive link, please let me know. I shall proceed further with the basic analysis. Advantage downloading SRE file is very fast. So FastQ file, I mean, there's no such as advantage over here because this is a publicly available database. We will get them in SRA files only. The files I have shared is I have converted them from SRA files to FASTQ files, and I was able to upload them in Google Drive for the workshop purpose. But in case you are downloading from any publicly available database like an SRA, you will have to convert them. Otherwise, if you have your own FASTQ files, that should also work. I'm repaste the link for the FASTQ files again. Uh, Please let me know once you are done with it. For tomorrow's analysis, you can maybe download one normal sample also, and such as over here, you can download the first sample is a normal sample. The one which I've uploaded is a patient sample, the test sample. So I would request you to download that. And for now, please only download the two files 98 546981 546982. These two FASTQ files shall be enough for now, so that we can quickly move on with the workshop. Uh, and meanwhile, while the raw data has been downloading, we can install FastQC tool. So the very basic command I will share here is This is the command you can use to install FastQC. I shall also share a link in case you need to refer to. You can always search for installation of FastQC in terminal, and we'll give a link. This is a very basic command you can use to install. So on one side, when you're downloading the data, you can also install this tool and keep it. I'm pretty sure many of them were able to install FastQC previously. And so if you have, in case everyone, if anyone is finished with the raw data and installation of FastQC, to analyze your file using FastQC, I'm pasting the command here. You have to just suppose you have two files or three files using .gz format, which are compressed file, that is .fastq.gz file names. You can directly use the command as fastqc *gz. So this will take into consideration all the fastq.gz compressed files. Make sure there are no other files in the folder, else it might end up throwing errors, or it will even consider all the other compressed files as well. And in case your FASTQ files are not compressed, I request you to please compress the files using the command gzip. So other command can be fastqc minus t6.gz, or you can give fastqc minus t. You can give any number of threads as per your system. It can also be 10 minus t, 10 also is possible. You can give your file name dot fastq.gz, etc. Any either of the way works. So it will process your FASTQ file, and it will give you two files, one HTML report file and one zip file, which is also the report for FastQC. Please let me know if anyone is stuck somewhere or I need to repeat some particular command. I'll keep. Please switch on your audio and ask, since I cannot change the screen every time for the commands. Uh, do you have a CentOS system to download, or you have a Windows system? Can you please specify which is a system?
I have an Ubuntu system.
So it's Ubuntu. You can always directly use Ubuntu, and in for people who are using terminal in Windows system, please use a Windows file. And for Mac, it has also specified a Mac file here, Mac OS X. That will be the file for your Mac. Uh, so based on your original Mac OS Big Sur, I don't think it has for the other one, but you can always use with the Mac OS X. That shall work, Miss Nathan. And this is the commands I'll keep. Is anyone facing problem with installation of fast QC tool? Or shall I proceed with installation of other tools? Or shall I wait? Please let me know. I shall be sharing all the commands and installations of the tools specifically for tomorrow's analysis anyways. But shall I proceed further or shall I wait? You can always message and let me know right now in the message chat
Ma'am,
box.
can you send this notepad stuff what you're typing in the notepad in the group, ma'am?
Yes, yes. I will do that, but I can do that after the workshop ends. Right now, I don't think I can. You want me to copy paste anything in the message box? I can do it right now. I'm just copy pasting this in the chat box. You all can always copy paste in your notepads so that you all can refer while you're doing it. Uh, now, if you have finished your fast QC, is it okay if I proceed further? Please let me know. Um, is everyone on the same page, or shall I wait for some more 5 minutes? I shall missing operand. Um, okay. Miss Aditi, maybe I can answer your questions after the workshop. I need to check with the system. Maybe you have some missing installations in your terminal. I can guide you maybe after the workshop, not a problem. In the meanwhile, it'll be better if you know if you can manage to download the FASTQ file and or you can just proceed further with FastQC analysis also instead of creating a directory because that may be a a permission error. So okay, I'll move forward. So once you have executed FastQC, it will usually give us result files in the form of one HTML file and one zip file. You can always open your zip file and see what are the results it gives you. It gives all your images, single fast camera image, per sequence based image, and it also gives an HTML page which is a club of all the results. So here, when you open your HTML file, you will see the statistics. The first one is basically summary statistics of your file. What is your file ID, file name, and then it's an Illumina based file. So its encoding is given as Illumina based and total number of sequences flagged as poor quality. In the sequence length, of course, it will be 150 because Illumina uses 150 base length only, and GC percentage here. Now the green ones are for those statistics which have passed the filter. That is, they are past the past QC filtration. The red ones, the crossed ones, are basically these parameters have not passed the filters. Like per base sequence content is not clearing the quality filter. Just a second. Yeah, downloads sometime they do take a while, but I suggest you to connect to a very fast internet if you have, or else I'll anyway share the logs for the analysis. You can keep it for download and you can follow them. I'll give you a detailed log as to how to follow for the analysis. You can execute the command as it is for tomorrow's workshop. But try having a single standard internet for this download. Otherwise, it might also end up with a corrupt file. We don't want that. And now here it's showing that the purse based sequence content has not passed the filter. So you can always take a call based on the graph here. In the y-axis, there are certain sequencing reads which are dropping down to 10, 20, and 30 scores as well. Fred scores, which is not a good sign. Then per GC content also, we can see the graph, which is very abrupt here. It should be a clear hyperbolic graph, but the red line gives you a weird, disturbing graph here. Then again, sequence duplication levels. There are many sequences. This indicates that there are many sequences which are duplicated quite a lot. Adapter content, it's not clear. It's because the raw data contains adapter sequences, which is accepted because this is a raw data directly taken from the Illumina sequencing machine. So in the second step, we have to this adapter content in the which has failed. In it also tells us that we have to proceed further with adapter removal, not just trimming the data. Adapter removal also, and KMA content is also so. When we proceed further with the low quality reads removal and adapter removal, what happens is high chance the failed filters also clear off because it removes all the adapters and low quality reads as well. So to move forward with the adapter removal, there's a tool called Trimomatic, or you can also use dbtools dot sh, which is another easy tool. You can just install either of them. I will share the link 1 second. So here I have shared links for down installing of BB tools and Trimomatic, both of them. Uh, I will show you with the Trimomatic tool, but and I will also share the commands for BB tools as well. BB tools is much easier to use, and Trimomatic is also easy. You just have to keep in mind what is the type of sequencing reads you're using, paired end or single end, and accordingly you have to make sure that you are giving the commands for trimming of the adapters and removal of low quality reads. So I hope you all can maybe you can try installing Thermomatic or BB tools, either of the following. BB tools doesn't take much time for installation. Is everyone online? I can give you 5 minutes, when you're just set up, so that I can show you the Trigonometric command as well. Just I can wait for 5 minutes to proceed. Just want everyone to be on the same page. Also, in the meanwhile, if anyone has any doubts, please let me know. I shall be able to answer you all. Shall I proceed with the trigonometric analysis, or shall I wait? BB Tools installation, you just have to unzip the file in the same folder you are working. It will be much easier for you, because for people who are new to Linux system, so that you can easily access the file and proceed it. And Trigonometric also. So for people who are working with Trigonometric, I can show you how it will be. Can I proceed? May I get a response? Or shall I wait more time? Hello? Sure. Okay. People who want to use Trimomatic tool, which is also a widely used tool for trimming and removal, can take a look at my screen. So here, usually Trimomatic file, it's a Java tool, based tool. So it will have a jar file. It's like you can see in the underlined here. It will have a Trimomatic jar file. So you need to give your path exactly the same way. So whenever you come execute a Java file, you have to first give Java jar space the path to the file. Like over here, the file is in this folder. I'll give this file, and you can press enter. Oh, gosh. So this will give me all the options the Trigonometric tool is using. So I have actually installed two versions for it, 0.36 and a current Trigonometric version. So now, because the files I'm using are parent files, I'll have to use both the files together. So here, if you can see the commands, it has given a command for parent. PE is parent, SE is single end. So whenever I'm using Trigonometric, I have to first give Java space hyphen jar space the path to the jar file. That is where exactly the Trigonometric jar file has been installed. And then I will proceed with the command. The first I will be using is PE. This is for parent. That indicates that my files are parent. Threads I can give 5. And Fred quality score I can select to be 33. Or it always takes default value. It can be anything. So default value is always 33, so you need not mention it. It's also fine. Then if you want a log file, it will print a trim log file, or else you can proceed further also. Then input file, it will be there. I'll be having two input files. So one will be one SRR forward file and one in reverse file. Then I have to specify the output file for it. That's minus O. Whatever, I can just give the same. You can give it as trim GZ. So uh, is everyone following the same?
Please ask the question in the chat box, or either someone has asked
Uh,
in WhatsApp group.
uh, yes,
Please ask the
yes.
question in chat box so that it will be easy. The question asked is by Atithi. Which way should I use to download Linux?
Linux. I would, if you are very new to downloading it, I would suggest you to use it in the Windows subsystem only. And otherwise, if you can manage to dual boot it, that is also a very good option. But for dual booting the system, you will need a pen drive which is of more than 8 GB, and you should format the pen drive and you have to make it a bootable device. Download the Linux folder in it, and you have to change the system when it is showing the grub options. So if you are well aware of the software the systems, you may have to do it. You can do it directly, or you may have to take someone's help. Yes, answering your question, input files are your read one and read two files, which like when if in case you have downloaded the files I have given, you will have _1 and _2 dot fastq files. So _1 is the first input file, that is the forward read file. _2 is the second input file, that is the reverse file, reverse read file. So you will need both the files, and output files also it will specify the same way. So um, you can also proceed without giving -o as yes, I will share all the links to the WhatsApp group once the workshop is done. And I can also try doing it now. One second.
Today's portions are over, Ms. Ishwari.
Uh, Doctor, no, I mean, I'm waiting for everyone's response. If they
Okay,
are
okay, okay, okay.
able to do it or not, accordingly, I can take a
Okay,
call.
okay. I can solve
Yes,
all the
yes.
problems of the
Yes,
participants,
yes.
then we can go. Okay.
Yeah, I am giving a command, the commands. Just a minute. I'm trying to put it in WhatsApp as well. So to install BB Tools, I'm giving the links in WhatsApp group. So here I have, you can copy paste this command I have shared here in the notepad, so that you can refer to this. This is one of the standard command for Trimomatic, which I will explain. So here they have used Trimomatic, and they have used PE, that is paired end, to specify that the reads a paired end. Then fret quality score has been given. Then read one file, read two files. So you need not worry about all the other files here. You have to specify your input file, that is read one file, read two file. If you have many, you can give all the file names with spaces in between, or you can just give one file again. And then coming out to Illumina clip contaminants forward reverse. So here that indicates that if you want to clip the Illumina adapters as well. Yeah, I'm actually currently not executing this command because I want everyone together. So I am sharing the standard command we use with Soundarya. Uh, just a second. Uh, Mr. Arun, the cutoff of Fred score actually you cannot. I mean, if you want, you can always increase it, but do not lower it one, and that you have to decide based on your studies. But usually, the default cutoff for Fred quality score is 34 or 33. All the tools are by default they consider it as 34 and 30 or 33, 34 itself. So you cannot worry. You need not worry about the Fred quality score. But again, it's all totally your call. If you want to adjust it, you can also adjust it. uh, what I will be doing is, based on the samples we are downloading today, we have downloaded today, I will share the logs for installation of the tools and how do we use the tools forward. I would want you all to just do one assignment, that is download or install either Bowtie 2 or BWAMEM for analysis, for alignment, and I will give the command. I will share the link as to from where to install and how to install, and you can just copy paste the commands as it is in your terminal. It will the tool gets installed, and you can give the second command for alignment as well. And once the alignment is done, I will also give the link for downloading the variant calling tool, so that for tomorrow's workshop we can work on uh, analyzing the variant calling tool, because these alignment and variant calling tool will require overnight procedure. So I would request you to do it today itself, by tonight. Installation does not take a lot of time for Botai 2 and BWMEM, so it should be done once the workshop gets over, and you can give it for alignment and variant calling. You can just copy paste the command as it is once I share, and you can so that you can analyze the result tomorrow. Then I will tomorrow anyways I will give a tutorial for alignment and variant calling, and we can proceed with annotation for the variants and visualization of variants. So I hope this is fine with everyone. I will share the detailed logs including each command and installation link. So that should not be a problem. And tomorrow I will go through the entire alignment and everything as well. So in case you missed out some problem or somewhere, we can always understand. So I hope you all can download the raw data. Yes, yes. I will share the commands log right away today, and I will give the detailed links of what all we have covered. And I will also share the presentation so that you can understand what topics we have discussed yesterday and today. And commands, you need not worry. I will definitely share them. Any doubts, any queries for today's session? Please let me know.
Ma'am, I need to access this WhatsApp group, and over there you would be sending a couple of links. I just
Yes.
need to follow those links and the instructions one by one after the other, right?
Yes, yes. I will give a log file with all the commands we use to analyze the files, and the installation commands and installation links also. So, you get an idea as to how do we install and how do we analyze the files as well. Because the further process is alignment, and then we have variant calling. So these two are very lengthy process, they take a very long time to perform. So for tomorrow's session, if you want to get the variant result file, I would suggest we to perform the alignment tonight itself. They take a long time, so I will give the command, you can just follow the command as it is.
Okay, I mean, if I'm able to follow whatever is to be followed today, then I
Yes,
should be able to do this alignment.
yes, yes.
Acha, okay.
I will give you the steps to download the this thing. Also, I will right now I will be showing how to download the reference genome before I
Okay.
conclude the session. So the reference genome download is a very easy step. So I will show you all how do we download the reference genome so
Hmm.
that it's ready for the alignment.
Okay.
Yes.
Thank you, ma'am.
So shall I like proceed further for everyone? I can show how do we download the reference genome, and then I will show you how do we you proceed further for alignment. Yes. One one second. Yeah. So for reference genome download, now one thing we need to keep in mind is the organism we are studying. The current For the current workshop, we have used Homo sapiens. The study uses Homo sapiens as the organism. So we need humans sequence reference genome. So what I will do again, I will go to NCBI genome, and because as I said, genome is a database which contains all the organisms sequenced genome, and I will search for Homo sapiens as it is. In case suppose I was using chicken like in the previous study, it is known as Gallus gallus. I will copy paste it as it is, and I will proceed further. For future analysis also, in case there are any scientific names we are not familiar with, we can always search them in the taxonomy browser, and or just Google it and you will get the scientific name as well. So here, as you can see this NCBI page, I have got a link to reference genome. So it gives me the reference genome. It is GRCH38P14. Uh, Sorry about that. It gives me the information about the reference genome, which one is currently uploaded. The current one is GRCH38.P14. This is the latest version of human. Then it gives me links to download in FASTA format, either genome, a transcript, or protein sequence. Then it gives me the genome annotation links, and I can also blast them. So in whole genome sequencing, the reference based, which we are doing reference based, we need a genome, entire genome in FASTA format. So I will click it in genome. Now there are again two ways. I can, yeah, either copy the link and paste it in terminal, or I can directly click it on the Windows and it will just directly download it for me. So what I will do is I will just copy the link and I'll download via terminal. You all can also follow the same way. Make sure there are no extra spaces, otherwise it will not read the file. So, so once the difference genome gets downloaded, I will use it to index in the alignment tool, which I will. I don't want to confuse you all. So before we proceed with the alignment, there is a process called indexing. Uh, so what indexing does is, I will mention here. So before we proceed with the alignment, we index a reference genome which we have downloaded. Indexing, what every alignment tool needs to have an indexed sequence of the reference genome for performing alignment. So indexing is it creates its own set of files, so that it can pick the coordinates and it can align your sample. The simple when we use a BWA alignment tool, once this is downloaded, I can show you. I just have to use BWA index and the reference genome name. I can show the sample command here. So as you can see the second picture, it shows I'm using BWA MEM. So there are two tools for alignment we can use, BWA MEM or Bowtie 2. Both of them will have to index the reference genome. So it's the same. You just have to give the index command. First will be the tool name, then you have to give the index command, and then you have to give the file name of the reference genome. Like for example, in my system, the file name will be saved as it is GCF underscore whatever underscore genomic.fa. So I'll have to index this file, the FASTA file. After I index it, it will create, so I can give the name any way I need. I have saved it in my system, so it will index in the same way. That it will create 7 files: reference underscore one, reference underscore two, reference underscore three, and then Botai2 will also save it in 7 types. So once the indexing is done, you can proceed further with the alignment. I will share the command, and I hope it will make it clear for you all to follow this. In this slide, the third image shows the alignment command with using BWA tool. I would not want to confuse you right now, but I hope you are following what exactly is happening in the analysis format. Please let me know. I hope you are following this, or should I repeat some part? Yeah, to download reference genome, again, I will show. You can always go to NCBI genome in Google, and you have to just type the scientific name for human. In this current workshop, we need Homo sapiens. This link, I will share the link. NCBI genome is a database which has downloaded, which has completely sequenced uh, sequence information of the genome information. But there are specific databases, like for Arabidopsis thaliana, there is a TAIR database. Then for plants, there is Plants Ensemble database. So you need to make a list of databases. You need to know which database has information about what organism, and what information about the organism. Accordingly, some people for plants especially, some people prefer an ensemble plants and not NCBI database for analysis, and especially for downloading the reference genome. So, based on that, for Homo sapiens, we can always use NCBI database. You can make a list of databases, for your references, and you can also find the reference list of databases in the book I have shared. So that shall help you guide you through the list of databases in bioinformatics. Initially, it might be a little tough, but I mean, you need to just to correlate with every analysis you do, and you shall be fine. I will also share the general flow chart right now. Uh, as you see this page, this gives you the general flow chart for WGS sequencing analysis. Like as I said, we will get some DNA samples, and they get sequenced either in Illumina, Ion Torrent, PacBio, or Oxford. The raw reads we are using is an Illumina sequencing raw reads. So what we have done is we have done the raw data fast QC control, quality control using fast QC, and then you can also use FastP tool, not a problem. I have used FastQC. Then a Trimomatic is a tool which will trim the data, or you can also use vbtools.sh to trim your data. So this will give you a very clean data with good quality scores only. It will remove all the low quality reads and adapter sequences from your reads. Then you proceed with mapping the reads, either using VWA or Botai2, and then you use Picard tools to mark your duplicates. And the other part, right from marking duplicates, variant calling, I shall be discussing this tomorrow because I don't want to load entire data. And I will share the link for this paper as well, so you can read it once. The paper describes in detail all the WGS analysis, the types of sequencing approaches used, everything. Any doubts about this session? Mr. Amitabh, the session is pre-recorded. Yes, a chat you can copy paste it and put it in your notepad. It can always be saved, and the links I shall always be pasting them in WhatsApp. Uh, the links, and if you want, I can also put the logs in Drive also, and I can share the Drive link, so you can always download it in our system. Yes, today I will be sharing all the links to download all the tools, and I will also give the logs to install them. And you can also use them for alignment and variant calling by today itself. So tomorrow we can understand the files and actually go forward with the analysis workshop. Any doubts, or shall I conclude the session?
I hope okay, some doubts are coming.
Yes, the files for the analysis is, as I had mentioned, you require raw data. You can download the FASTQ files from the drive link I have given, or you can also follow the steps I have shared initially from SRA database, then convert them to FASTQ files. I have shared it in the command message chat box also. You can follow them, or you can directly download the FASTQ files I have provided from the drive link, whichever suits you. But I will be sharing the entire log, so you can always follow it.
Hope many people have some doubts yesterday. Today, Miss Ishwari has given an extra day for taking the class because many people said about the basics, all these things. So, hope today it was she has started from the basics and it was worthy for you. So,
Any doubts, please feel free to note it down, and you can always ask in tomorrow's session. I'll be
yes,
happy to answer them all.
yes, many
And I
people.
will share all links and logs so that you can always practice them after the workshop also. Yes, alignment and everything would be better if you could do it tonight itself, so that for tomorrow you can have a ready file. Even if you don't have, it's okay. I shall be having a file, so I can show you. But it will be better if you can also have a result file for a productive session. Okay, then I shall see you all tomorrow, and
Okay,
I shall share
we,
all the links before
we can wind up.
tonight.
Dr. Ishwari, we can
Yes, yes,
wind up. Okay. Thank you
yes,
everyone.
yes,
Thank you.
yes.
Tomorrow the same time
Thank you so
I
much.
will send the link.
Yes,
Tomorrow the same time we can join. Okay.